nostalgia. Habrá de ser un tango nuestra herida, un acordeón sangriento nuestras almas.
Welcome back to Your Brain, a Personal Tour. Today is Seminar 2, The Decision to Act. Whereas some, like Hamlet, had it easy and only had to make a decision about whether to revenge his father or not, in everyday life, the decision to act is much more complicated. Today, we're going to talk about the 4 ways your brain makes a decision to act. Some are automatic, some are very deliberative. Some are decided by Mother Nature, some are decided by mental time travel. We'll cover each today in this lecture. I begin with some background on one of my favorite quotes about deciding to act. This is from Amelia Earhart, and she said, "The most difficult thing is the decision to act. The rest is merely tenacity." This woman, dubbed Queen of the Air, was the first female soloist across the Atlantic. She was a best-selling book author, married to a famous book publisher, George Putnam, and a strong advocate for equal rights. Fierce determination marked Amelia once she had made a decision. That's why she's on this slide today. She tragically died in 1937 on a circumnavigational flight around the planet due to a memory problem. But memory's the next seminar. We'll talk about decisions right now. While Hamlet had it easy, life presents us with everyday demands that require decisions, and they begin at a very early age. Let's take a look at one example.
The marshmallow test is a really great way to show how children delay gratification. We tried it out with the 4 children we've been following since September 2010, Alfie, Millie, Mackay, and Pratmesh. Here's how it works. We had each child on their own sit at the table at a desk with a plate and 1 marshmallow. They could either choose to eat the one marshmallow right then and there, or they could wait until I came back into the room and have 2 marshmallows. I left them alone in the room for 15 minutes. Take a look. The marshmallow test has been used for decades by psychologists. It's been used with children to predict later academic success, including literacy, SAT scores, and other academic outcomes. There's no definitive answers from the marshmallow test. It's not a matter of passing or failing. What we're looking for is whether children can really resist this piece of white candy sitting in front of them that's sweet, that, the smell of it, the allure of the marshmallow. In Pratmesh's case, we really saw this added curiosity because he had never actually tasted a marshmallow before. All of the children managed to show some level of self-control and resist the temptation to eat the whole marshmallow. As you can see from the footage, you can catch a glimpse into children's ability to control their impulses. This ability, which is developed around the time of kindergarten, can be linked to other outcomes later in life. At the end, the marshmallows were in kind of different states. Some had been squished, ripped apart, nibbled around. There was this temptation and there was this impulse to kind of try it out.
In this tape and in many discussions, this test is presented as a delay of gratification or an exercise of self-control. I think once you see what the children actually did, you'll see that they're deciding not to eat the marshmallow by deciding to do something else. Thus, much of the socialization, much of the choices we are presented in life are not delays of gratification or managing our self-control. It's deciding to do one set of actions rather than a given action. There are 4 ways, as we said, that your brain accomplishes that. Let's look at each one in turn. The first kind of decision is a reflex. It's a decision that Mother Nature makes for you. because this decision is too important for your brain to decide. It's a stereotypical behavior, typically of defensive nature. It's designed to protect you, to keep you alive, and therefore much of the action, which is really a basic building block of movement, is decided before your brain even gets involved. A good example is the withdrawal reflex. Now, this reflex comes into play whenever one of your limbs hits a painful stimulus. Let's take the legs in this instance. So here in this diagram, you've just stepped on something sharp or you've stubbed your toe, something like that. There's specialized receptors for that, called nociceptors. They're at the end of a very long nerve path where the actual neurons, the cell bodies, are in your spinal cord or very close to your spinal cord. That detection then comes into the sensory neuron, which then decides to tell your brain, but that's going to take a long time, relatively speaking. And then immediately a set of actions are brought into play. First, muscles are both activated and relaxed so that you can bring your foot away from this sharp and painful stimulus. But notice what also happens. Your other leg is then prepared to handle the shift in weight. This is a fairly complex behavior, and it is pre-wired by Mother Nature in your spinal cord. Your brain is informed, but you're going to feel, quote, the pain way long after you've moved your limbs, your legs in this instance, to accommodate this stimulus. Mother Nature is good for you, and this is a good example. I want to draw your attention to a special class of neurons called interneurons. Let's look at a different reflex because I think this is simpler to show the point I want to make about interneurons. Let's suppose you've decided to put your hand in a particular position. What you've really done is specify what that muscle spindle shown in this diagram in your bicep should be experiencing in terms of tension. If something were to fall in your hand, that muscle spindle would elongate. It would send a message into your spinal cord, and then a reflex would occur to actually tighten, to contract the bicep, to keep your hand in the same position, but also to relax the antagonist muscle. The interneuron, which is the little gray neuron shown in between this sensory motor loop, is an inhibitory interneuron. It can cause motor neurons to not get excited, to cause a muscle to relax. But it also points to another role of interneurons, and that is they can be used to override reflexes. This is an additional layer of control. It's our first instance that reflexes, while stereotypical, can also be overridden. And a good example of that comes from a very famous scene in a very famous movie.
What would care if you did? Allow me to ignite your cigarette.
Sir? Mr. Lawrence?
Yes?
Flimsy, sir.
Thank you.
You do that once too often. It's only flesh and blood.
Michael George Hartley, you're a philosopher.
And you're balmy. Oh! It damn well hurts.
Certainly it hurts.
But what's the trick then?
The trick, William Potter, is not minding that it hurts.
So each of us has an inter-Lawrence. We don't have to go around in flowing robes to override our reflexes. But the other side of this equation is our brain still is involved. It still is informed that, yes, indeed, putting out a match with your fingers does hurt. But we have the capability, by using higher order circuits in our brain, to override even the most basic reflex and building blocks of movement granted to us by Mother Nature. That is important as we explore more of this wonderful organ called the brain. So let's bring Homer back and ask him to summarize the two fundamental points we want to make about reflexes, the first decision we've studied in this seminar. And the first point is, a reflex is a do-it-now decision. Your brain is involved after the fact. There's neurons in your spinal cord to handle this kind of sensory detection and movement immediately. But because there's interneurons, each of us do have an inner Lawrence that can override these reflex actions based on control from the brain. Having done that, let's look at our second decision. The second type of decision we talk about today are associative decisions. Now, these decisions unleash a reflex-like behavior, but from a learned association. An association that has typically taken a benign stimulus, something like a light or a tone, and paired it with a stimulus that elicits an autonomic or reflex-like behavior. This kind of learned association is uniquely associated with Ivan Petrovich Pavlov, who in a conditioning paradigm often termed classical conditioning or Pavlovian conditioning, took dogs, measured their saliva response to the presentation of food, and then preceded that delivery of food with something called a conditioned stimulus. That was often a light that blinked on before the food was delivered or a tone. You can see the apparatus here. This would be at the turn of the century in the early 1900s, a bit more primitive than it is today, but still Pavlov elucidated a kind of learning and a kind of decision that is still studied to this day. However, today we don't study Pavlov's dogs, and I must note that my wife Becky is a professional dog trainer, so I know she'll be very proud of me for getting a dog into these slides. The dog really of research today is the California sea hare. You see two of them crawling on the ocean floor today. These animals have very simple behaviors. They basically move around in the sand. They squish water out of something called a siphon. You'll see it disturb the surface of the water here. And they have a gill to breathe. These animals have turned out to be the perfect research animals for associative decisions or Pavlovian or classical conditioning. Now, the scientific name for this California sea hare is Aplysia californica. And since he's from California, we're going to give him some cool sunglasses that you can see here on the bottom left. But really, he's cool for one unique aspect in his brain. The California sea hare's brain has got relatively few cells, 20,000 cells or so in the entire brain of this animal, compared to 86 billion in your brain. But the unique feature of the Aplysia brain is that the neurons themselves are visible to the naked eye. If you look in the center section, the neuron R2 is over 1 millimeter in diameter. It is roughly the size of the B in a Liberty penny. So these individual neurons can be identified visually and then studied. So that means we can take, multiple different California sea hares, and within each we make a small incision. We can now study its brain, but we can identify uniquely the R2 neuron in each and every individual Aplysia brain. That means we can study the neuronal basis of associative decisions in the California sea hare. Now let's take this dude and teach him a new trick using associative learning or Pavlovian conditioning. And what that means is we're going to take a previously benign tactile stimulus, so that's noted up here in this diagram, and we're just going to basically touch the animal. Normally, nothing would happen. But what we're going to do is pair that touch, or the CS, with an unconditioned stimulus of shock. So we'll shock mildly the animal's tail. When we shock the animal, there's a vigorous and strong withdrawal of the siphon, that part of the animal that expels water. And what we'll find is after we train the animal by pairing this tactile conditioned stimulus of touch to the shock unconditioned stimulus, that after training, there's a vigorous withdrawal of the siphon at the presentation of the tactile stimulus. When we started, this tactile stimulus didn't do anything. When we finish associative conditioning, the tactile stimulus will withdraw that siphon. And that's shown in the bottom right. After training, you see a dramatic increase in the amount the siphon withdraws to the conditioned stimulus, and this indicates learning. Now, the nice thing about aplesia, the reason we chose this animal, is that the brain neurons that mediate this effect, the entire process is underpinned by 3 neurons. There's a sensory neuron that detects the CS, this tactile stimulation. There's another neuron, actually this neuron has a number, L29, that detects the US. The L29 neuron on its own can activate a motor neuron that withdraws this siphon, and that we see after training, the sensory neuron, in addition to the L29 neuron, now can both activate the motor neuron to withdraw the siphon. Now let's look in detail at the changes in the brain of the aplesia as it learns this task and a new decision pathway is created. We're going to look at the sensory neuron and the L29 neuron's juncture with this motor neuron. Remember, the sensory neuron detects the touch, the L29 neuron detects the shock, the motor neuron is the one that produces the behavior, the withdrawal of the siphon. I want to expand it out, and then just to be sure, this sensory neuron is depicted along here, so that's that sensory neuron. This motor neuron is depicted just right here. When we begin the training, the sensory neuron cannot evoke the motor neuron to contract the siphon, to produce any kind of behavior. And that is because there's insufficient neurotransmitter shown here in these little packets. There's insufficient neurotransmitter being dumped onto the motor neuron from the sensory neuron prior to training. Once we begin training, then the first of 2 changes occur. There's a short-term change where previously empty legs of the sensory neuron now become filled with additional packets of neurotransmitter, and now all of a sudden the motor neuron is affected by the sensory neuron and the siphon begins to withdraw. If we continue this training, then over the course of several hours, about 12 to 18 hours in this instance, a dramatic event happens. And that is, there's actual growth in the brain of this animal. New legs appear on the sensory neuron. They become populated with neurotransmitter, and now there's a long-term structural change in the brain that reflects this learning and will now evoke the decision to withdraw the siphon to show this learned effect, and it's because there's structural changes in the brain. These are long-lasting structural changes and underpin, if you will, the memory of this training. But right now, we're just focusing on the fact that when this tactile stimulus is now presented, there is a decision based on these changes in the brain that will drive the motor neuron to action and show the learned behavior. These same changes that happen in aplesia, the see here, also happen in other organisms like rats, like us. And those kinds of changes are important to understand how emotional conditioning might happen. Let's take an example with a rat. If you were to take a rat, place him in an environment and give that rat a shock, what you would see her doing is freezing. That's what a rat does when threatened. She gets a foot shock, she freezes. The pathway in the brain for that foot shock is shown on the right. It goes through a couple of areas of your brain and then concentrates into a lateral area of a point in the brain called the amygdala. Now, the amygdala is a specialized structure in mammalian brains that is kind of dedicated into detecting items that are threatening to the organism, or very, very good to the organism, but we'll stay with threatening right now. And there's two parts to the amygdala. One is the lateral nucleus, that's designated LA on this slide, and one is the central nucleus, that's designated CE on this slide. But when that rat gets a foot shock, basically there's a path to your brain into the amygdala. The amygdala says freeze, and it does that by the lateral nucleus causing the central nucleus to fan out as part of the fight or flight syndrome, and you get the external behavior of freezing that is shown on the bottom. But freezing isn't the only thing the rat does when it gets shocked. There's also internal changes to the rat. The blood pressure of the rat goes up. Her stress hormones are released into her system. So one point I want to make about this is that when unconditioned stimuli, like foot shock, dramatic stimuli, they elicit a behavior externally that you can see, like freezing, but they also elicit internal body states, rises in blood pressure, release of hormones. If we now associate a tone with that foot shock, we will see associative conditioning, Pavlovian conditioning, and now all of a sudden the tone through the amygdala, that's where the pairing happens, that's where the changes like we just saw with the aplesia would happen. Now all of a sudden the tone will elicit the external behavior. But the tone is also eliciting the changes in internal state. The tone will now elicit blood pressure rising. The tone will now elicit stress hormones being released. And this is extremely important for us to understand how emotional conditioning will be happening along with behavioral conditioning. Let me give you an example from humans. In this example, it's almost twilight, you're walking in the woods, and you run into this. There's a lot of learning that went into your upbringing, at least in the West, that says any tree that looks like this is capable of chasing you, grabbing you, and doing things you don't want to think about. So this is a threatening stimulus. Your amygdala is going to go crazy. And one of the things it will do as it prepares to orchestrate this fight or flight syndrome is your blood pressure is going to go up. Stress hormones are going to be released into your bloodstream. Your heart is going to race. And based upon a long set of psychological research, then because of those things, because of your blood pressure going up, because of your stress hormones being released into the bloodstream, and because your heart is racing, you will report that you feel fear. We feel fear not because we see this thing and then all of a sudden, our blood pressure goes up and our stress hormones get released. We see this thing, it's detected, internal changes in our body happen, like your racing heart, like your blood pressure going up, and then we feel the emotion. Stated another way, we feel fear not because we're looking at the bear, but because we're running from the bear. So this whole set of associative decisions is part and parcel of how we learn emotional responses to various stimuluses. These emotional responses are automatic. They come as part and parcel of the associative conditioning process. And that is one way that emotions get attached to various things in our life. This concept of the source of emotion can be used for treatments other than scaring people in the woods. It turns out about 20% of the troops returning from Afghanistan or Iraq suffer from PTSD. Standard therapies only result in about 60% remission. The great challenge are members of the special forces who report hypervigilance and anxiety, similar to the effects shown in the movie American Sniper. Well, if you block a specific autonomic nerve center, called a ganglion, with a pain blocker, there are widespread reports of immediate and profound relief. One shot seems to block the emotion-generating aspect of higher heart rate, fast breathing, and muscle tension. In other words, blocking the physical attributes that underlie emotion seem to release the perception of stress and allow therapies to progress. But brain-related changes as a result of stress don't just affect combat veterans. It hits children living in poverty as well. In fact, home life in many such situations may approach a combat situation. And using the techniques developed for PTSD and underpinned by emerging brain science offer real hope for new pathways out of poverty by changing behavior. Here, the emphasis on minimizing the oft-reported chaos in the environment with stress-reducing exercises, as well as exercises on making better decisions. Measurements show that the stress response of the body reduces, brain health improves, and better behavior results. This creates a positive cycle of behavior, termed better deliberative decisions. We'll study them in just a moment. So this approach then is therefore a real cost-effective path to help alleviate some of the impacts of poverty on a young person's brain and life. So it's time for Homer to weigh in again. We are now going to ask Homer what he thinks about this whole set of associative decisions. And Homer has 2 points to make. One is, even slimy sea thingies can learn to predict the future. We take an innocuous stimulus like a touch and pair it with a shock, and even a California sea hare can predict the future based on that tactile stimulation. It will do something in anticipation of that shock coming, and that is an associative decision. The second thing Homer wants to say is those kinds of associative decisions change your brain, and it changes your brain with new growth. That's what's the structural change here as you learn things. Now this has several consequences. Your brain, as we know, is fully encased with bone. That means it's a fixed volume. If every time you learn something there's new growth in your brain, sooner or later it's going to run out of space. How do we deal with that? How does the brain deal with that? It turns out that is one of the prime functions of sleep. But we're going to learn about that later. Right now, we're going to go to the most complex decision making your brain can do. These kinds of decisions are called deliberative decisions. They're decisions where we have to trade off priorities or options. We could do one thing or the other, and we think about that, evaluating this before we actually decide and commit a particular behavior to action. Oftentimes, these decisions are our most memorable. We remember agonizing over these kinds of decisions. We recall what we were doing and how we came to a certain decision. And believe it or not, even our little friend like the rat here goes through the same thing. What we'll see is that decisions as they're being made, deliberative decisions as they're being made, your brain and our friend the rat brain here goes through what can be thought of as mental time travel. I know that sounds fairly sophisticated for a rat, but we'll see that both our brain and the rat's brain have structures that help us perform those kinds of activities. To really get a good handle on this, we're going to have to look at 3 different places in the brain. So let's take one by one. The first brain area I want to talk about is called the hippocampus. We see it here in a rotating model of your brain. It's the highlighted linear structure at the base of each hemisphere. It's about the size of your little finger. And it's amazing that a part of your brain the size of your little finger is so important in the way you make decisions, in the operation of memory, etc. It is really quite amazing. The hippocampus came to prominence when this young man, at the age of 27 in 1953, received an operation to cure his epilepsy. The operation, which removed his hippocampi, both sides, and parts of his medial temporal lobe, so there was more than just his hippocampi removed here, was very successful at curing his epilepsy. But it came at great cost. In essence, this person, while he was alive, he was known as H.M. He died in early 2000s, and we now know his real name was Henry Meliasen. Was able to really form and convert no new memories about his experiences to long-term memory. He was well aware of his memories of childhood, but every time he met his doctor, he had to be reintroduced. He would remember the doctor's name during the course of the session, so within 30 minutes to an hour he could interact and he was fine. But the next day, when he met this doctor, and he'd met the doctor many times because H.M. was a celebrity in brain research, he had to be reintroduced. Now it turns out H.M. could make memories of a different sort. He could learn drawing and coloring tasks, so he had procedural memory or motor memory. But taking memories about his experience, something called episodic memory, and converting them into long-term memories and making decisions about them was way beyond his capabilities. So this brought the hippocampus, this brought HM into the crosshairs of intensive brain research. Now, the hippocampus had been known to neuroanatomists since the 16th century. In fact, the first guy that described the hippocampus actually at first thought it looked like a silkworm. The hippocampus from a human brain being shown on the left here, and a nice little cuddly seahorse being shown on the right. The anatomist changed his mind and said, after further inspection, it does look like a seahorse, so being a good neuroanatomist, he translated this all into Greek, and voila, we get a new name for a part of the brain, hippocampus, literally translated as sea monster, but again, most people think seahorses are cute little creatures, and so we'll go with the positive aspects of the name, and that is why the hippocampus is called the hippocampus. So as the hippocampus came into prominence in brain research, people began to experiment with rats and rabbits and monkeys and everything, the dynamics of specialized cells within the hippocampus. And one of the most important discoveries that is really fairly recent is the discovery of hippocampal place cells. Now these are cells, this is behavior of cells within the hippocampus that after a rat has explored some space, let's take this S-shaped track here, all of a sudden you find like yellow cells and orange cells and green cells and red cells, etc., that seem to express when the rat is at a particular point in space. So for example, if we could record the neurons in this rat's brain and we let him run down this track, when he got to this yellow section of track, we could see some yellow cells. They're not really yellow, but I'm going to tag them this way. And when the rat got to that point of the track, the neurons would do this. So they would burst. Then later on, when the rat got to the orange part of the track, a different set of cells would do this. So notice, there would be populations of cells within the rat's hippocampus that would preferentially burst when the rat was at a particular place on the track. These cells, they're not being grown like what we talked about with aplesia. They change their behavior, so they arise after the rat has explored this space, and then they activate when the rat is in very particular places along that path. I assert When the yellow cells are firing here, the rat is thinking about being in the yellow portion of this maze. That's this area up here. When the orange cells are firing, the rat is thinking about being in the orange portion of this maze. Now that is amazing, and let's see it in real time. What we are seeing, or going to see, is actual recordings of an experiment where a rat is exploring an M-shaped elevated walkway in an actual research lab. The position of the rat is going to be denoted by a white light here. He's connected to a wire that allows us to record and amplify the neuronal signals. We're recording from 10 individual cells, and this is a major advance compared to when I was in graduate school in the 70s. When I was a young graduate student with lots of hair, you could record maybe 1 or 2 cells and you considered yourself pretty lucky. Now, neuroscientists can record from dozens of cells, and because we can get dozens of cells recorded all at the same time, we can look at their interactions as an animal explores space, makes a decision, or performs some task. The firing of those cells will be denoted on the left-hand side of the screen. We'll put a little vertical marker, a little line, whenever that particular cell fires, and the cells are color-coded so that we can map the activity of the cells against where the rat is in space. Now let's see what happens. I'll let the film loop here a couple of times. I draw your attention to the following. Note how the rat is going first up and then left and then down. That's his path through space as he negotiates this elevated walkway. The cells follow a particular pattern. The blue cells fire first, then the kind of greenish ones, then the yellow ones, then a few reds and pinks when he finally finishes. So this rat is walking in space through a path, up, then left, then down. We can do that by following the white light. We can also do that by specifying the colors in sequence of how the hippocampal place cells fire. First blue, then greenish, then yellow, then red, then when he finishes, pink. That's another way of describing the path that this rat is taking through space. So it is the relationship of these place cells, the sequence with which they're firing, that describes the path through space that this rat is taking. Hippocampal place cells do not just fire as the rat is physically exploring space. They also fire at other times, and that's going to help us understand how a rat, quote, thinks about a path. The other time that hippocampal place cells fire are during something called a sharp wave ripple. Now, this is a very short event. It's only 1/10 of a second long. But during those sharp wave ripples, we see very interesting behavior from place cells, and that's shown at the bottom of this slide. During a sharp wave ripple, as recorded from this rat, this is abstracting experimental data, we see place cells firing in sequence. Purple, blue, green, yellow, pinkish, brown. Now we know place cells actually map to space. So if we reconstructed the actual places in a maze tied to those colors of place cells, we would actually trace out a path. If you decode that path, it's shown on the right. Now the rat is not physically moving through space. This happens much too quickly. It's 1/10 of a second long. So what is happening here, and I truly believe this, is that the rat is thinking about going down in this maze, turning right, and then turning right again and going up. We are actually showing now, with experimental results, these have been recorded, a rat thinking about moving himself through time, through space, and this is what we would call mental time travel on the part of a rat. The second area of the brain we need to talk about is a time-traveling reward system. And that's what we're looking at in terms of this slide. Now, the reward system of your brain is actually tied to a specific neurotransmitter. Neurotransmitters are those chemicals that one neuron dumps out on another in order to influence its behavior. There's several different chemicals, but the reward system of mammalian brains, so your brain, the rat brain we've been looking at and what have you, uses a specific neurotransmitter called dopamine. So oftentimes this reward system is called the dopamine system. Now, what do I mean that this reward system travels in time? We'll look at 3 different events. In the first event, represented by the neuronal histogram up at the top. So this is kind of a record of brain neurons in the dopamine system and how they're firing as things happen. There'll be a light that randomly comes on, and there'll be food that's dumped into a rat's cage that randomly gets dumped in, and we'll call that the reward. You'll notice that the dopamine system doesn't do anything to the light, but boy does it go crazy when a reward is dumped into the cell. So remember, your brain has no sensors, no feeling, it can't decode sensory data, it can't, and so it needs an internal representation of reward, and the dopamine system is exactly that. And here's a good piece of evidence that when you put in a reward for a rat, which is a piece of food, for example, the dopamine system goes crazy, and that's great. It also works with anti-rewards like shock and those kind of things. So this truly is stimuli of salience to the animal, but we're just going to stick with reward right now to show our point. Now, the middle tracing is when we have paired a trigger with that reward. So we're now into a Pavlovian or classical conditioning scenario. It's what we talked about with associative decisions. So there's a trigger that predicts the reward. Notice what's happening in this central trace on the slide. The dopamine system migrates in time and now reacts to the earliest event in time that predicts the reward, in this case, the triggering stimulus. It goes off on the trigger, and note that it doesn't go off on the reward. The reward is totally predicted by the trigger, so it moves in time to now go off on the trigger. On the bottom tracing, we've actually taken this a step further. We now have an instructional signal, something that basically comes on to tell the animal, oh, by the way, the CS is going to come on next, which will then be followed by the US, the reward. And look what the reward system does. It goes off again to the earliest predicting signal, in this instance, the quote, instructional cue, that then predicts the trigger, which then predicts the reward. And it doesn't go off on the trigger or the reward anymore. You'll note that the vigor of the response decreases over time. The best thing for the reward system is the actual reward. But something that predicts it, or something that predicts something that predicts it, will also cause the reward system to express itself, but in increasingly smaller and smaller doses. So here is a system of your brain that can represent reward, but can also travel in time. Much like the place cells represent mental time travel of space, the dopamine system can represent mental time travel of reward. The third and final part of the brain we need to talk about, before we put it all back together, is something called the basal ganglia. Now, in this rotating X-ray kind of view of your head, the basal ganglia are the colored bits, the various colored bits that sit at the base of the brain. There are multiple individual nuclei there. We don't need to go into them right now. We'll just consider them as a whole in terms of the basal ganglia. And the way I want you to think about the basal ganglia is that they are a go, no-go system of the brain. Various potential movements or behaviors are in a race condition in the basal ganglia. The first one that gets to a threshold gets committed to behavior, and the other ones are suppressed. Until that happens, there's just competing potential. No real behavior happens. Now, all sorts of behaviors are going on at all sorts of times, so it's not like the animal is completely cessation of moving, but the behavior under deliberation is going through a race, if you will. The first one, in terms of potential movements that get to threshold, is the one that's going to be committed to behavior, and let's see how that works. Let's look at an example taken from real experimental data. We'll consider two potential actions, turning right and turning left. And we're going to look at a slightly different metric of neuronal behavior. This is called a heat map. So the more neurons that are firing and active and excited will be depicted by warmer and warmer colors. So more reds, more yellows, but especially, deep reds mean lots of neurons are firing. Cooler colors like blues mean less neurons are firing. Now in this diagram shown on the right-hand part of the slide, many neurons are shown. Some in the basal ganglia, this go-no-go system, are associated with turning right. Some are associated with turning left. Each trace of what a neuron is doing is aligned with the arrow that points with time. So there's probably 100 neurons depicted here, and we're following them in time as two things happen. The first is both potential actions are shown early on. When the animal gets close to a choice point, both actions are represented within the go-no-go system. We see that right here on the diagram. Then something happens. One of the actions crosses a threshold. Once it crosses that, then that potential action is actually committed to behavior that's going to be expressed. Now we've got to get the movement object positioned, signals have got to be sent out to the muscles, so it will be some time before the actual movement occurs. But the prime point I want to show with this slide is potential actions are all simultaneously active. One gets to a threshold before the other, and then that's the potential action that's going to be committed to express behavior. Hence the deeper reds, hence the higher peaks of the mountain. That's the movement the animal is going to do. I draw your attention to what happens to the other potential movement once the threshold for action is reached. It goes very blue. That means that potential action is being suppressed. And that is a hallmark of this system. One of the potential actions hits a threshold, it's committed to eventual behavior. The other actions are suppressed. This will be important when we talk about obsessive compulsive behaviors, where one action is chosen by somebody to do over and over again in order to suppress the emotions associated with a different action. But let's just keep it simple right now. The main points here are multiple actions being represented, hit a threshold for action, that's the one committed to behavior, the other actions are suppressed. Now let's put this all together. Let's put all of this together and watch as one of our favorite rats move down to a choice point in a T-maze. Now this particular rat, this is a female rat, I can tell, she will go down, and she's been trained in the past that the green path, i.e. turning right, actually is the direction of food. So food has been consistently paired with turning to the right. No food has been paired with turning to the left. That's the reddish path in here. And this is a trained mouse. So what is going through her brain as she gets close to the choice point? Well, what's happening is that her hippocampus, using the place cells and with these sharp wave ripples, these are 1/10 of a second long thoughts, if you will, get her to mentally travel down that path, turn right, and then there's been reward associated with the end of that path. The reward system, which can also travel in time, evaluates that. She also, on the next sharp wave ripple, will mentally time travel down the left or reddish path. No reward has been associated with that. The reward system's not very active. Now, this gets repeated again and again. Almost accumulating evidence is one way to think about it. The basal ganglia, as a group, now is associating or evaluating turning right versus turning left. Early on, they both race to a threshold. The reward system adds more evidence, more processing, if you will, to turning right, because that's the choice that has been associated with reward. Turning right gets to the threshold first. That then gets committed to expressed behavior. Ultimately, a movement will occur. Muscles will point her towards the right. She'll scamper down that right-hand part of the T-maze, and the left-hand turn will be suppressed. Once that threshold is hit, and before the behavior is expressed, she is no longer thinking about turning left. She is thinking and preparing to turn right, and that is the committed behavior. Note, think about that. We have now seen her think, deliberate over various paths, make a decision that will then get expressed into behavior. Isn't that awesome? Let's see some of these cells in actual operation during the course of a real experiment. This is a rat in a self-returning maze. She will run down the center part of this maze and then make a right or left turn. She then comes back to the top, runs down the path again, and makes a different turn. The rat has been taught to alternate. It first goes right and then goes left. That's the behavior. And we'll look at some cells that show how the rat is deciding to do that in real time. Experiments that I won't show you show the rat have ruled out that these are just alternating cells or that they're head direction cells, etc. The point of this is I want to see if you can use the actual behavior of the cells to predict which way the rat is going to turn. Listening to those cells, I hope you reach the conclusion that they were active and bursting when the rat was going to go left, not right. So the manipulations, the behavior of these cells that we've talked about leading up to a rat making this choice point is actually expressed in real live brains of real live animals when they're making a decision. So it's time for Homer to join us again and talk about the two main points of this section on deliberative decisions. And Homer, in his own unique way, will say that even rats have a brain that can travel in time. This turns out to be of fundamental importance in the way to understand the brain. Our brain can move us around in time. A rat's brain can move them around in time, both for physical space, for reward. That can then feed other systems that can, decide via race conditions which particular potential action is actually going to be expressed in the movements of muscles and the repositioning of the animal in space. But Homer also has a second point, and if you know anything about Homer, you know he's a bit of a lazy guy. And what he says is, but it sure seems to take a lot of effort into making this kind of decision. And he is very, very right there. Your brain takes 25% of your caloric intake. In other words, 25% of the energy in your body is spent on processing of the brain. And Mother Nature is very careful about how you spend energy, and we can't keep that up all the time. And so there is a fourth kind of decision that is motivated from this kind of discussion. Habits. Habits are decisions that your brain has delegated and really not much engaged in anymore. They occupy an amazing amount of our time, and they are quite useful. Almost half of the behaviors that you do on any given day are habit. I bet you didn't know, but there are psychologists that measure how we spend our day in behavioral routines, and their research comes in at 45% of the behaviors we do on any given day is habit in nature. Some animals, the percentage is even more. If you don't think you're a creature of habit, try changing your shower routine. This is a highly stereotypical behavior for most of us, each behavior being cued by the behavior before us. We tend to have a very rigid routine when we do this. Or as we drive to work, tie our shoes, perform certain activities during the course of the day, we are all creatures of habit. Now, the benefit of habit is reduced energy load on the brain. How the reduced energy load on the brain is effected is related to another aspect of the reward system. And that aspect is the reward system is sensitive to surprise. We've talked about if you drop a reward into a cage of an animal randomly without prediction, it just comes in from the sky, no predictive event precedes it, the reward system gets accelerated. It accelerates its firing. So that reward system detects the fact that something significant has happened, wasn't predicted, neuronal rates go up, and we see the increase in the histogram as shown in the diagram in the top right of this slide. Let's, though, pair that reward with a predicting stimulus that's showed in the middle, and we'll see that, yes, the reward, the dopamine system accelerates its response to the stimulus that predicts the reward. That's right after the CS in this diagram. But when the reward is actually given, the reward system doesn't react at all. It keeps firing along at a steady pace. There was nothing surprising here. The reward was predicted. Therefore, it's keep on doing what you're doing. Now, suppose we predict an award with a conditioned stimulus, so there's a signal that says a reward is about to come, and then no reward happens. In this instance, the reward system, the dopamine neurons, cease firing. They decelerate. They detect this event. It was surprising. There was an expectation of reward. Nothing happens, and the reward system decelerates. So this has implications on the overall level of activity in your brain. That is because when your reward system is changing, either accelerating from its normal rate of firing or decelerating from your normal rate of firing, your brain lights up. It is very active when your reward system is surprised, shall we say. Now, the origin of the reward system is shown in the lower right. It is in your brainstem, the ancient part of your brain that controls breathing, autonomic functions, the things that you don't have to pay attention to at all. And then it innervates wide-ranging parts of the front part of your brain, the frontal cortex, and these are where outcomes are evaluated. Is this something I want to happen again, or is this something I don't want to happen again? So when your reward system is surprised, it activates huge portions of your neocortex, as shown in this diagram. Your brain is chewing up energy like crazy. That's good when you can't predict what is going on in the environment. But when you can predict what is going on in the environment, you can use a different strategy. You can use the simplest set of of stimuli in the environment that tell you what to do, kind of like your shower routine. You just use the simple movements that are occurring to predict what are the next movements you can make. And we can actually test for this experimentally. On the next slide, we'll talk about a famous experiment in the formation of habits. It deals with a rat in a maze where we will think about the maze as labeled with compass points. But before we describe the experiment, I want to make sure you have a practical view of the maze itself. It looks like this. Note, the rat can see all sorts of stuff in the room, and the rat could use them as signposts in making a turn. So from the rat's perspective, the rat may not be turning to sterile points of the compass like west or east. The rat could be turning towards the red chair or some other landmark in the room, and that's important. So let's see what happens in the experiment. The most straightforward way to test this is to take a rat and use what is called a cross maze. Now we train the rat in a maze that has 4 arms. It's shown in the left-hand portion of this diagram under A. We'll label the ends of each arm with compass points. So there's north, south, east, and west. We always start the rat from the south part of the maze, and the food is always in the east part of the maze. Now, being good scientists, periodically we're going to probe by changing up the situation on the rat, and we're going to start him on the north side of the maze. The rat can use 2 strategies, really, in deciding which way to go on the probe trials. If he has a play strategy, if he knows which way is east, then he's going to turn left when started in the north, and he'll go to the east, the fixed point in space where the food has always been. If the rat is using what is called a response strategy, he's going to do whatever the response he's done most of the time in training. Well, most of the time he's turned right. So if we probe him by starting him from the north, he's going to turn right and wind up going to the west. Now, if we do this, what happens? Well, what we find is if we probe early in training, when the rat is just initially learning, he's showing that he knows the food is in the east by turning to the right. So early in training, we probe by starting him from the north. We find, lo and behold, he takes a place-oriented decision. Now, that's a deliberative decision, and the rat runs to the east. Why? Because early in training, his reward system has got his brain lit up, his hippocampus is moving him around in time, the basal ganglia is integrating information, the frontal cortex is evaluating outcomes. This rat is aware of where the east is in this training maze, fully aware and processing all the stimuli in the room and the context, etc. And so that rat will go to a specific point in space, that is, the east. If we then overtrain the rat, we keep putting him in the south, he runs down, turns right, goes to the east, we overtraining him on this, then it becomes almost automatic, and now we probe. What we find is that late in training, the rat takes an associative decision. Turning right has been paired with the food so many times, the rat is not thinking about this. He's running down, he gets to an intersection, and the normal thing to do is turn right. It's a habit, and that's what he does. It turns out that at this stage of training, the rat is really only using a small part of his brain as part of the basal ganglia, the caudate nucleus, that's shown in green here. But the point is, he's not really processing many stimuli other than running down the maze means I turn right, I get to food. In this instance, it's not a very good strategy. But from an energy perspective and from showing us how habits form, this is exactly what happens in the formation of habits. We go from where our brain has been awoken and lit up by surprising events in the environment. We're fully engaged to understand that. We are cognizant of all the stimuli around us. And then as we overtrain on a task or we get much more familiar on a task, really we're only using a small part of our brain to figure out what to do next. As beneficial as habits might be for the energy utilization in your brain, they're also the source of much human misery. In fact, recent scientific research have shown perhaps much misery in gerbils as well. Because habits once formed, since they involve so little of your brain, are very, very difficult to break. Your brain is on autopilot, if it will. And when the habits are maladaptive, when their behaviors really that you shouldn't be doing, then it becomes very, very difficult to break these chains of autopilot behavior because your brain is really not evaluating other options. It's really not deliberating anymore. It's performing automatic associative decision where one step leads to the next, and at the end of this chain, it's not a good place. So Homer is going to weigh in at the end of this section and give us his two observations of note. And the first is, Homer likes habits. They help his simple brain not get too tired. Homer is not one to really apply himself most of the time, so Homer is absolutely a creature of habit. He manages his brain at a very low energy level, and that's just fine for Homer. We wish him well with that. But Homer also notes, once bad behaviors turn into habits, they are rigid and then therefore very difficult to change. This is one of the challenges as we address some of the problems in modern behavior, and I think Homer's got it nailed here, that the study of habits and the way of breaking habits can offer great hope to segments of our population that really need it. I'd like to close our seminar today with two overriding thoughts. The first is, we've talked about 4 decision systems. 3 of them, reflex, associative, and habit, are automatic. Your brains are wired to do this. Yes, you can override reflexes. There is some ways of modifying habit, but in general, these are automatic. They reflect the way your brains are wired. When ultimate behaviors get produced out of these systems, it's not necessarily because people are evil, it's because their brains are wired that way. So to make progress, we've got to mentally shift. We've got to get the clutch in. We've got to choose another gear to help us deal with potentially bad side effects of some of these decision systems. And that's where I think the deliberation system comes in. Deliberation is hard, but it can be used to address automatic bias and other kinds of automatic associations that our brains naturally make. A good example comes from modern symphonies. One way your brain has an automatic bias is that people generally tend to pick groups that are similar to themselves. So if white males choose symphony players, white males are going to tend to pick white male performers for that symphony. Modern symphonies have found that blind auditions accommodate and address the automatic bias of such a situation. Performers audition behind a curtain. Therefore, the evaluators don't know the gender, don't know the race, don't know the ethnicity of the performer, and this has turned out to be a wonderful way of eliminating gender and other kinds of bias in modern symphony selection. There are many other ways that the deliberation system can be used to address automatic biases in situations today. And then the second point I want to make is deliberation is rooted in space. We saw how a rat brain moves that rat around in space mentally. Your brain has the same circuits. So as you deliberate a particularly hard problem, as you consider options, etc., think about moving yourself around in space. There's an excellent example that comes from the time of Romans and Greeks called the method of loci. And this is the method to help people remember a large number of items. What they do is they position the items in space that is familiar to them. A very famous memonist put items he was trying to remember on various positions down his home village road. And when he wanted to recall these items, he took a literal walk down this road in his mind. This is an unusual aspect of deliberation. It doesn't a priori jump out at you, but I have certainly found that when I use the metaphor of moving around in space, it does help me make deliberative decisions. Before we finish this recording, let me foreshadow the topic of Seminar 3, the structure of memory. Memories are complex, multidimensional, slippery concepts that have several processes within the brain. They also have a structure within the brain. We'll talk about what are the essential characteristics of memory. One type of memory that has been talked about, at least in literature, comes from Proust. This French writer wrote 7 volumes and thousands of pages of an involuntary memory that came from a fateful encounter of a famous Madeleine and a cup of tea on a cold winter's day. The memories came in rich, detailed patterns, flowing out with, involuntarily into a rich remembrance of time long past. Is that the way memory is structured? Or is there another way? Well, Pacific Northwest Indians have a different concept, and they're based on totem poles. Totem poles are not an elaborate reconstruction of a story as told in the pages of the book. They're the essential elements of the story as carved on a totem pole. To give a familiar Western example, the totem pole of Goldilocks and the Three Bears would be a pole that has a bear carved in it, a bowl of porridge carved in it, and a bed carved in it. The story would be reconstructed from there. Now, of course, that means that 2 different people might tell the story 2 different ways, or the same person on 2 different occasions might tell the story 2 different ways, and we'll see whether memory structures relate themselves to that, like the ways of the Pacific Northwest Indians, or like Proust, or something in between. Please join us next time for the structure of memory.
Llégame, mata mi corazón. En nuestra alcoba azul